Morning children, and welcome to biology class. Today you are going to discuss about the second chapter in genetics and evolution, which is the molecular basis of inheritance. First we are going to study the structure of DNA. Children, what is DNA? I'm not asking the full form. You know the full form as deoxyribonucleic acid. Okay? Now what is DNA, children? It is considered as a long polymer of nucleotide. What is that? A polymer of nucleotide or deoxyribonucleotide. Clear? Okay. Now, what is one nucleotide? What is one nucleotide made up of now? It is made up of a nitrogenous base, a pentose sugar, and a phosphate molecule. It is made up of three components mainly. What are they, children? It is made up of a nitrogenous base, a sugar molecule, a 5-carbon sugar molecule, and a phosphate molecule. These three components make up a nucleotide, one nucleotide. Right, like that, many nucleotides are joined together to form a polymer of polynucleotide. Okay, right? So what are they? What is this nitrogenous base? There are two types of nitrogenous base in our cells, which are the purines and pyrimidines. What are they? Purines and pyrimidines. Based on the difference in structures, they are differentiated as purines and pyrimidines. And there are two examples of purines, which are adenine and guanine. What is that? Adenine and guanine. And three examples of pyrimidines, which are cytosine, thymine, and uracil. Clear? So, these are the nitrogenous base. When, for example, in this nucleotide, one nucleotide, a thymine, yes, thymine is a pyrimidine, right? Pyrimidine combines with a sugar molecule, 5-carbon sugar molecule. Clear? When a nitrogenous base combines with the 5OH of the sugar molecule to form a nucleoside. What is that? It forms a nucleoside. Children, once again, nitrogenous base, when it combines with the sugar molecule, it forms a nucleoside. Clear? Now you know this thymine. When it becomes a nucleoside, it is called as thymidine or deoxyribotymidine. Understood, children? Okay. Same way, if it is adenine, if in this place it is adenine, what will happen? Adenine is the nitrogenous base, right? When it combines with the sugar, it forms a nucleoside. Clear? And what do you call now? Deoxyadenosine. Clear? Same way, each and every nitrogenous base combines with a sugar molecule to form its respective nucleoside. Clear with me? Okay. Now, when this nucleoside combines with the phosphate molecule, it becomes a nucleotide. Clear, children? So, a nucleoside plus one phosphate group gives a nucleotide. Clear? Children, do you know what is the bond between a nitrogenous base and a and a pentose sugar. When a nitrogenous base bonds with the sugar molecule, 5-carbon sugar molecule, by a N-glycosidic linkage or N-glycosidic bond. What is that? N-glycosidic linkage or bond. Okay. To form a, what is this one? Nucleoside. Okay, a nucleoside. Okay, and now when this nucleoside combines with a phosphate molecule, and what is this bond, do you know? Which is called as phosphoester bond. Phosphoester bond. Okay, now this nitrogenous base combines with the pentose sugar through N-glycosidic linkage, and when this phosphate molecule joins to the sugar molecule at the fifth position, see, fifth position through a, what is the linkage? Phosphoester bond or linkage. Clear, children? Like this is one nucleotide. One nucleotide is formed. Clear? Same way, many nucleotides are formed. You know that. But what is the connection between, what is the linkage between one nucleotide and the other? It is called as phosphodiester. Phosphodiester bond or linkage. Clear? Okay. And what is the full form of RNA? How is RNA different from DNA, children? RNA and DNA is somewhat same, except that RNA has, in each nucleotide, has an additional OH group. Has an additional OH group at second position of this ribose sugar. What it has? It has a OH group. Every nucleotide has one OH group in addition at the second position of the sugar molecule. Clear? And another difference between RNA and DNA is, what is that? RNA is single-stranded, whereas DNA is double-stranded. Clear? See this one, OH. Clear, children? Okay. Children, do you know who proposed the structure of DNA? Yeah, it was James Watson and Francis Crick. Right. Even before these two persons, in 1869, around the time of Mendel's itself, Friedrich Miescher, who was that? Friedrich Miescher identified an acid from the nucleus, which he named it as nuclein. He identified or separated, he could not separate it, that's why. He identified a acid from inside the nucleus, which he named it as nuclein. Okay, but it was such a long polymer that he could not isolate it. Okay, and after nearly hundreds of years, in 1953, James Watson and Francis Crick, with the help of X-ray diffraction technique proposed by Maurice Wilkins and Rosalind Franklin, okay, use these to propose the structure of the very famous DNA model. Okay, for this they were awarded the Nobel Prize. Clear, children? Okay. Children, now you know that DNA is a long polymer of nucleotide, isn't it? Nucleotide. Okay. On one end, there is a free phosphate molecule. What is there? Free phosphate molecule at the fifth end of the 5-carbon sugar, fifth position. Okay, and what is the other end? What is the end of the other end? They have a, what is that? OH molecule. OH molecule at the third position. Okay, right? So we also call it as the polarity, or we call it as 5 prime and 3 prime. On a polynucleotide chain, or the polynucleotide, this is called the polarity of the polynucleotide chain. What is the polarity, children? We call it as 5 dash 3 dash or 3 dash 5 dash, whatever it is. Clear? We call it as 5 dash and 3 dash. Why this 5 dash 3 dash, children? On one end of the polynucleotide chain, we have a phosphate molecule at the 5th carbon position of the sugar molecule. Okay, and at the other end we have a OH molecule at the 3rd position of the carbon. Clear? That is why it is called as 5 dash 3 dash. Clear? Which is called the polarity. Now you know that the DNA is not a single-stranded molecule. It is made up of a double-stranded molecule. Clear? Okay. And you know, both the strands are polynucleotide strands. Okay. Now if there is a purine base on one of the strand, for example, adenine on one of the nucleotide of one strand, definitely on the other strand it will be a pyrimidine. What should be the pyrimidine? It should be the thymine. A always base pairs with thymidine. Okay, and if the next nucleotide has one purine, what will be the other? It will have the pyrimidine called as cytosine. Okay, now what is uracil there? You know there is one more type of pyrimidine, uracil. What is uracil? Yeah, uracil is seen in the case of RNA in the place of thymine. Right, actually thymine is 5-methyl uracil. Clear? Okay, right. So if on one of the strand, if there is a purine, on the other strand, definitely there will be a pyrimidine. Okay, and also you should know that A, that is adenine, always base pair with thymine, and G, that is guanine, always base pairs with cytosine. Okay, it should not, it can be otherwise also. It can be T and A, not only A and T and G, C always. It can be TA, it can be CG, like that. Anyway, it can be. Okay, right. And also you should know that what is the bond between these two base pairs? This is the hydrogen bond. Hydrogen bond between these base pairs. And also you should know that between adenine and thymine there are 2 hydrogen bonds, and between G and C there are 3 hydrogen bonds. Clear, children? 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between G and C. Like that, you know, 2 strands of polynucleotide is formed. Okay, one of the strand has the polarity if it is 5 dash 3 dash. The other strand has definitely the polarity 5 dash 3 dash, or they have the opposite polarity. Okay, children, clear? So children, DNA is made up of 2 polynucleotide chains coiled in a right-handed fashion. They are coiled around each other in a right-handed fashion, children. Okay, and in each of these coils you can find nearly 10 base pairs. Nearly 10 base pairs. Nearly 10. Okay, and this is called as the pitch. Okay, between each the distance between the pitch is nearly 3.4 nanometer. How much is that? 3.4 nanometer. And the distance between the two base pairs, adjacent base pairs, is 0.34 nanometer. Okay, right. And the entire length of DNA is supposed to be 34 nanometer. Okay, clear? Now you know that the nucleotides are, of course, or indeed the base pairs, made up of base pairs, right? So we can call the DNA molecule as a polymer of nucleotides or a polymer of base pairs. Okay, also you should know that if there are 100 adenine molecule, there should be definitely 100 thymine. Yes or no? Yes. If there are 50 number of G guanine, there will be definitely 50 number of cytosine. Correct? But is it necessary that the adenine and guanine or cytosine and the thymine should be the same? No. But adenine plus guanine is equal to thymine plus cytosine. Or A plus G divided by T plus C is always equal to 1. Okay, which was given by, this equation was given by Erwin Chergaff. Who was that? Erwin Chergaff. So this rule also helped James Watson and Francis Crick to propose the very famous model of DNA double helix. Now it's called as DNA double helix. Okay, it is coiled upon one another in a right-handed fashion. Clear, children?